thank you for the introduction. I am really honored to be here today to be part of this Women in Data Science conference. And I want to thank the organizers for thinking of me when they were organizing. So I'm going to talk today about discrimination, classification, same idea, sample groups with multi-way biochemical neuroimaging data. Here are some acknowledgments. So this is joint work by a PhD student in biostatistics, Tianmeng Liu, and a colleague of mine on the faculty, Eric Locke. And then we have several collaborators who provided the data that we worked with at the Center for Magnetic Resonance Research, Gulen Ersh, Dinesh Dilshan, and Uzay Amir. And the data come from a particular group of patients who have a disease called Spinocerebellar ataxia, and they are recruited through Khalaf Bouchar's clinic here on campus. There are two NIH funding sources here. One is an R01 that funded the patient study, and the other is a KL2, a training grant that supported my colleague Eric Locke. And we have no conflicts of interest to report. So I'm going to spend a little more time on background than I might usually because I wasn't quite sure who would be in the audience. So first I want to tell you what all the things in my title mean. So what groups am I talking about today? I'm talking about ataxia patients versus similar aged healthy controls. By biochemical neuroimaging, I mean in particular magnetic resonance spectroscopy. I'm going to tell you what I mean by multi-way data. That's a term that may not be used very widely outside of the little niche where we use it. And I'm going to tell you just a little bit of background on classifiers. I think probably many of you are familiar with some classifiers, but in case there aren't, I'll cover that briefly. Then the main focus of the talk is going to be on a particular classification procedure called distance-weighted discrimination. And what we're going to do is talk about an algorithm we developed to adapt DWD to this scenario of multi-way data. And I will show some performance properties of the algorithm and then apply it to the ataxia data set. Okay, so what is spinocerebellar ataxia type 1? There are something like 15 different types of ataxias. They are all dominant hereditary polyglutamine repeat diseases. They cause dysfunction and loss of Purkinje cells, causes brain atrophy. Symptoms are loss of coordination and balance. That's often the first symptom that people come up with and what leads to their diagnosis. As the disease progresses, it involves speech and swallowing difficulties and muscle stiffness. And the typical lifespan is about 20 years after symptoms first appear. If symptoms first appear relatively young, late teens, early 20s, that often means a more aggressive disease and death can occur sooner. If it's later onset, often means a slightly longer lifespan. So MR proton spectroscopy. So this is just a little bit of intuition about why MR works to get at biochemicals. You've probably heard enough about functional MRI and structural MRI to have an idea about that, but this is a little bit different. So in particular, it is proton spectroscopy. You can also do sodium spectroscopy and phosphorus spectroscopy. So in this case, hydrogen's one electron orbits, like the Niels Bohr model of the atom. But the proton also has angular momentum, and that is called spin, and that creates an inherent magnetic charge. But now those hydrogen atoms are embedded in molecules, and so the surrounding electron cloud of all of the atoms around the hydrogen in that molecule means that the protons resonate at slightly different frequencies depending on the surrounding cloud, whatever molecule they're in. So this is what the MR scanner can detect, those slightly different resonances depending on whether hydrogen is embedded in glucose or hydrogen is embedded in ascorbate or hydrogen is embedded in N-acetyl aspartate. So the image, which in this case is not really an image the way you usually think for MR, is a spectrum. And I'll show you a picture of a spectrum. So here on the left-hand side, we have a structural scan of a control. And the little yellow box shows the region where the biochemicals are being captured. And on the right-hand side, we have a patient. And this is a spectrum from one person at one time. This is a spectrum from the control at that one time. The horizontal axis represents the resonance frequency. So that tells me which biochemical I'm looking at. It's known, for example, that NAA resonates at 2. So we know that the peak that appears at 2 corresponds to NAA. And then how much NAA is there is quantified in the peak. So what do I mean by multi-way data? What I mean by that are data that have multiple dimensions, a particular structure, specifically multiple dimensions. So here are a couple examples. In my ataxia context, we have a number of different biochemicals measured in each of 3 different brain regions. We have another data set that I'm not going to talk about today where we have gene expression quantified for several genes longitudinally over time in a sample of multiple sclerosis patients. An example that's a little more engineering oriented, perhaps we have measures of degradation in a bridge structure at each of several time points. So for each bridge that we've sampled, we have locations by time points. And I'm going to use the notation here, dimension 1 by dimension 2. So most statistical and machine learning techniques would take what is inherently a data matrix, where I have each of my many biochemicals in each of my 3 brain regions, and vectorize it. Just stack all the values on top of each other, and then ignore the underlying structure. Would not pay attention to it at all. So maybe that's okay. Why do I care? In the ataxia example, maybe the biochemical pattern that's informing the classification I'm going to do is actually pretty similar for some of the brain regions, or all of the brain regions. So it should be to my advantage to have the algorithm know what that underlying structure is. There may be examples where that underlying structure doesn't actually inform, but there are likely to be examples where it does. So here's some examples of classification approaches. Some of the older ones, Fisher's linear discriminant analysis and logistic regression. Some nonlinear examples, k-nearest neighbor and random forests. And then 2 more modern linear approaches that I'm going to focus on today, support vector machines and distance-weighted discrimination. So what I like to have in a classifier is an approach that produces weights or coefficients, measures of how strong a particular variable is in informing the classification. So I want coefficients that indicate the size and the direction that that variable makes in its contribution to the classification. For these linear procedures, then also, if I take those weights, those coefficients, in a linear combination with the data, my variables, then I get an underlying latent continuum, and that is, in fact, what informs the classification. We threshold on that to decide, is this particular sample predicted to be a case or a control, or a bridge that is degrading or not degrading? Okay, so linear classification in particular identifies a high-dimensional hyperplane, a linear combination of my predictors, my variables, and coefficients that best separates 2 classes. There are procedures, such as DWD, where you can have multiple classes, not just 2. But today I'm going to focus on 2. So I'm going to call my classes the +1 group and the -1 group. So support vector machines maximize the distance between the class +1 and the class -1 samples that are closest to the hyperplane. Those are the samples that are hardest to tell apart, right? Because they're very close to the hyperplane. Maybe they should be on that side of the plane, maybe they should be on that side of the plane. They're the hardest to tell apart. So that's what SVM focuses on. That introduces a problem that's called data piling, which you may have heard of if you used SVM. And so DWD avoids that data problem by instead allowing all of the samples to affect the estimation of the hyperplane. And both of these allow for big data extensions, what statisticians call the P larger than N problem. I have more variables than I have samples. The example I'm going to talk about today is not a big data problem in that context, but these are easily applied to a context like that. So I have N samples, which in my ataxia example, yeah. Let me take it off my sweater. Okay. So I have n samples, which in my ataxia context are people. I have n people. Each person I know belongs to one of the two classes. These are patient or control. This is not a clustering problem where I don't know the underlying classes. This is a classification problem. I know the classes. I have a vector of predictors for each sample. Those are my biochemicals in all of the regions. I have a class label, and there's just a notation for setting up vectors and matrices based on those values. So my goal is to find this coefficient vector B that defines the hyperplane that best separates the two classes. How do we do that? We have to set up an objective function, and that objective function has to be optimized in some way. And the exact form of the objective function h and any additional parameters it depends on, tuning parameters, penalty parameters, things like that, depend on which method we use. DWD uses second-order cone programming to optimize the penalized sum of the inverse distances from the data points to the hyperplane. So this piece is the penalty, and this piece is the distance to the hyperplane, what statisticians would think of as a residual. And the residuals are defined with these constraints. So we have this objective function. We optimize it. We get our estimated coefficients. And then this is the equation that defines our hyperplane. And then we threshold this to assign a prediction. Is this particular sample a case or control according to the prediction? So we're going to define this as the full model because I have as many coefficients as I have combinations of metabolites and regions. I've done no dimension reduction yet. Okay, so my coefficient matrix is of size p times n. P is the dimension of my first dimension, and m is the dimension of my second dimension. So why are we calling that naive? Because we think we might be able to do some effective data reduction, dimension reduction. So how might that help us? It might make sense for the context, but it also might result in better classification. If I have too many parameters and they're not all contributing to the classification, I should try to weed some out. So our proposed approach, specifically to take account of the underlying multi-way data, is a low-rank approximation of that big coefficient matrix B. So we start with a rank 1 decomposition. So we assume that our coefficient matrix B can be decomposed into the outer product of two vectors, W and V. W is going to denote the vector of weights or coefficients for my dimension 1 predictors. V is going to denote the vector of weights or coefficients for my dimension 2 predictors. And the hyperplane to separate the two classes is now defined like this. And so you can probably see here where my algorithm is going to go. So I can take this piece as if it were a data piece by itself and find the W's. Then I can take my W's, pretend these are my data pieces, and find a V and go back and forth. Okay, so, but there's an assumption here, down here at the bottom. So this assumes that the importance of different predictors in one dimension is proportional across the levels of the other dimension. So if I have a particular pattern across the biochemicals in brain region 1 that informs the classification, proportionately, the other brain regions have a similar pattern weighted up or weighted down to inform the classification.
So do you actually find that to be true? I mean, the brain, in terms of the metabolites and the neurotransmitters it's using, are highly sort of selected to where you are in the brain.
Yeah.
There are chemicals that are only expressed in the pons that aren't expressed elsewhere.
Yeah, it's certainly not going to be true in every context. I'll get back to the example at the end of the talk. Okay, so here's my iteration. So we take a previously estimated vector v, treat this now as our input to the algorithm, and get our output estimated weight vector w. Then we take that w, treat this product here now as the input to the algorithm in order to get the output, a new estimated updated vector v. Okay, so we have to worry about whether or not we can actually get this to converge. So the objective functions of DWD and SVM and other linear classifiers are convex. But now I'm iterating between two separate implementations of DWD. This is one fitting. This is another fitting. So this is an example of a biconvex optimization problem, and it should work. So does it work? So we carried out a simulation experiment to compare the full model to our proposed rank 1 multi-way model. And in fact, we considered other low rank approximations also. So we generated data randomly under several conditions. We had two different sample sizes. We kept them fairly modest sample sizes here in order to look at small sample properties. We considered different dimensions for the array. This is very analogous to our ataxia example. This is getting a little bit towards big data, not huge, but big. And then we simulated data under different structural forms distinguishing the two classes from each other. So we generated some data where the truth was the full model, where I really need that full P by M matrix of coefficients to do the classification. And we also generated data when the rank 1 model was the truth and when the rank 2 model was the truth. So what's a rank 2 model? So I think about my ataxia example. A rank 2 model means, for example, that a particular biochemical pattern informs the classification in one way for some of the regions, but there's a different classification pattern across the biochemicals in other regions. Okay, the model was fit on a training set and then tested on a test set. Each scenario was replicated 200 times independently. And we used a package that's already out there called DWD to do the fitting. So first piece, is the algorithm valid? And what I mean by that is, if we feed into the algorithm data of a particular type, does the algorithm tell us back the right type? Does the algorithm tell us, yes, that was rank 1 data, I got it? So yes, the model with the best misclassification rates was the true model, the model from which the data were generated in all of those scenarios, the full and the low rank situations. And as we expected, when the size of the training sample increased from 40 to 100, the misclassification rates got better, lower. And when the dimensions increased, the differences between the true model and models that weren't the truth got bigger, right? The other models failed more as the dimensions increased. So the problem of fitting the wrong model got bigger as the dimensions got bigger. Okay, so here's just a simple graph, a piece of some of the simulations that we did that illustrates this. So you can see when the full model is the truth, the full model has the lowest misclassification rate. When the rank 1 model is the truth, it has the lowest misclassification rate. And when the rank 2 model is the truth, it has the lowest misclassification rate. Here are some higher order, higher rank models. And this is how the general rank R model is expressed. And we see the same pattern holds. When the rank model that is the truth has the best misclassification rate consistently across the different types of models. Okay, so now a different question. Does this outperform other classification procedures? So we compared the proposed rank 1 DWD model to other linear and nonlinear classifiers, which were designed for high-dimensional classification: ElasticNet, DWD, L1 norm, DWD, L1 norm, SVM, and random forest. And consistently, when the rank 1 model is the truth, the rank 1 DWD model has the lowest average misclassification errors under all of those sample size and dimension scenarios. And again, the misclassification gets even better as the dimensions increase. So as our rank 1 model, in some sense, becomes more strongly true, the rank 1 fitting does better and better. Whoops, I'm missing the last piece of my table here. Sorry about this. So this is just a table illustrating that. So here, these are the misclassification rates averaged across 200 independent realizations. We have the sample size and the dimensions, and you can see that the rank 1 here, this is all rank 1 is the truth. All the simulations here are rank 1 is the truth. So these are all fitting full rank models using different algorithms. And so the misclassification rate is the smallest when we're fitting the correct model. So none of that is surprising. If we didn't see that, we would be very worried. Okay, so back to the ataxia problem. So we had 17 patients and 24 controls. Each person was imaged in a 3T scanner with a spectroscopy voxel placed in each of the pons, the cerebellar vermis and the cerebellar hemisphere, spanning the midline. And you can see, if you've worked with any MR data, these are very big voxels compared to functional MRI and structural MRI, for example. So in each region, we had, I think this is 14 different neurochemicals: ascorbate, gamma-aminobutyric acid, glucose, glutamate, glutathione, myoinositol, siloinositol, N-acetyl aspartate, total choline, total creatine, total NAA, which is NAA plus NAG, glutamate plus glutamine, and glucose plus taurine. Now, we wanted to quantify the misclassification in a way that allowed us to avoid the problem of overfitting. If we just fit our model to the data once and we get one misclassification rate, that's overly optimistic. So we used 10-fold cross-validation, 41 subjects randomly partitioned into 10 subgroups. In each step of the cross-validation, one of those subgroups was left out. The model was built with the remaining 9 subgroups and then tested on the left-out group. And then we rotate so that each subgroup is left out once. And then we repeated that 100 times with different random partitions into those 10 subgroups. This is a fairly small sample situation, which is why we did that. We didn't want to have generated, for example, an initial partition into 10 subgroups that was odd in some way. Okay, so results here. Average misclassification rates were 7% for the rank 1 model, 4.5 for the full model, so that answers your question about this particular data set. 8.5 for elastic net DWD, 9.3 for L1 norm DWD, 10.5 for L1 norm SVM, and 6.9 for random forest. So there wasn't compelling evidence for this data set that the rank 1 model was really the best. The DWD did a little bit better. Now, that difference, 7% to 4.5%, is actually due to one person. This is a small enough data set that one model misclassifying one more person really changes that percent misclassification. So in particular, in this data set, we had 2 patients who were called pre-manifest. So they know they have the disease because they've had it in their family and they've had their genetic screening. So they know they have it, but they don't have any symptoms yet. So they look a little bit more like the controls than like the patients. So it's a data set where there are 2 people that are hard to classify. They're just different. Okay. Does that answer your question? Yeah. Okay, so here's just a picture. You sometimes see these pictures when people want to illustrate the data piling problem for support vector machines. So here I'm just illustrating there's no data piling problem. So the score down here is the X prime B, the calculation using the fitted weights from the model. The vertical axis is arbitrary. It just shows a density. This is one of those pre-symptomatic patients. And the other one is, I think, this one that's quite close to the dividing line. So the dividing line, the hyperplane, is here at 0. Okay, inference. Statisticians care about inference, and what I mean by that is I don't just want to know the coefficients. I want an estimate of how variable they are. I want to know if coefficients are strongly different from 0 or not. Okay, so we have a weight vector for our biochemicals, and we have a weight vector for our regions. To estimate the 95% confidence intervals of those weights, we used 5,000 bootstrap samples. And here's what we get. So the biochemicals here are ordered by the magnitude of their coefficients, from strongly positive to strongly negative. And here are the region coefficients. We can see that the coefficient for the pons has a little bit larger of an area than for vermis and cerebellar hemispheres. The pons is a more difficult region to image, and there is some atrophy in the brain in the patients, which makes it even a little more difficult. Up here in the biochemicals, we have NAA and total NAA and inositol and total creatine and glucose plus taurine showing as strong informers of the classification. So is that believable? The biochemicals with the large absolute weights and with the confidence intervals that did not cover 0 are those that have been shown in other studies to distinguish patients from controls. Inositol is a glial marker. Creatine is related to energetics in the brain, as is glucose. Taurine is an organic osmolyte. Glutamate is a neurotransmitter. Glutamine is a precursor to glutamate. And NAA is a neuronal marker. And I already mentioned this. So what do we conclude in general about this low-rank approach? So compared to naive applications of a full-rank model, linear or nonlinear, multi-way DWD showed similar or better classification performance and an easier interpretation in the context. Our framework to extend linear classification to multi-way data is not specific to DWD. You could take the same sort of approach with other classifiers, and we've already implemented it in SVM, for example. We focused on DWD because of its ability to overcome the data piling of SVM. So where do we go next with this? We just finished a longitudinal study in the same cohort, so now we have biochemicals by regions by time points. So a word of caution, of course. While the simple rank 1 model performed well in situations where the rank 1 model is the truth, when you're sitting at a computer with a data set in front of you, you never know what the truth is. So it's always good practice to try several ranks, all the way up to full rank, and use some principled model selection approach to try to decide which model best fits the data. We have an R package available on GitHub, and this is in a paper that is forthcoming in Biostatistics. Thank you very much.